This talk is about how our capabilities as surgeons has tracked advances in visualization, not only from technological innovations, but also through improved artistic techniques that help communicate our specialized knowledge. Visualization has been a core mystery of our existence, occupying many of the greatest thinkers all the way back to antiquity. Plato and most other prominent thinkers before him, such as Pythagoras, believed that rays emitted from the eye mediated vision. Some speculate that this began with the observation that light seemed to be present inside the eyes of animals lit by torch at night. They didn't yet understand reflections from the retina. The theory was also a bit problematic because it meant that these rays would have to have infinite velocity, because we have an instant view of the moon and stars when we open our eyes. Plato's most famous student, Aristotle, disagreed with him and argued for a more empirical approach to the science of vision. Democritus and Epicurus, who were prominent competitors to Plato, argued that effigies of an object were received into the eye, rather than the object being touched by rays emitted from the eye. But nonetheless, Plato prevailed. Working mainly in Alexandria, a city founded by Aristotle's most famous pupil, the mathematician Euclid encoded Plato's ideas into a formal mathematical theory of optics, in which a cone of rays emitted from the eye traveled in straight lines to interact with objects, much like an extended sense of touch. About a century after Plato, the first medical schools of the ancient world were forming in Alexandria. Here, the ancient father of anatomy, Herophilus, described the lens, and thinkers began to speculate that it was the seat of vision. Several centuries later, also in Alexandria, the great Roman physician Galen built upon Plato and Herophilus and incorporated his ideas into his theory of pneuma, a vital substance originating from within the cerebral ventricles that, in the case of vision, flowed through hollow optic nerves into a spherical lens located at the center of the eye, where it produced visualization with the visual rays. As the main surviving text containing the wisdom of the Alexandrian school of medicine, Galen's writings remained dogma for over a millennium. In the Arab world, where ancient Greek knowledge was being kept alive during the Dark Ages, scientists, through empirical observation, finally proved that vision occurred when light was reflected from an object and then into the eye. But in the West, it was not until the Renaissance that the dogma of Galen was challenged and scientific progress resumed. During this period, Vesalius showed that the optic nerve was indeed not hollow, as Galen had said. Felix Platter determined that the retina was the true sensory feature of the eye, not the lens. Johannes Kepler then explained how light passing through the lens could form an image on the retina, but it was still a mystery how this led to vision. At the dawn of the Enlightenment, Isaac Newton extended these theories by proposing that miniature vibrations within the retina were transmitted through the optic nerve to the brain to produce vision. He also proved that white light could be separated into component colors using a prism, and then the colors could be mixed as desired to create other colors. Then, in the 19th century, scientists deduced that the eye contains three types of color receptors, loosely related to red, green, and blue, that when combined in various proportions could produce all perceived colors. Their trichromatic theory of color vision greatly simplified the capture and reproduction of color images because the required ingredients, such as filters, film emulsions, inks, and emitters, would only be needed for three colors. Also in the 19th century, around the time photography was being invented, Charles Wheatstone was able to explain binocular vision and create the first stereoscopic images. This challenge had eluded earlier great minds, such as Leonardo da Vinci, who wanted to create images that leap from the page in 3 dimensions. But the details of how Newton's theory about the physiology of vision might work did not become clear until the 20th century, with the work of Hubel and Wiesel in the 1960s and 70s. They used microelectrode recordings in the cat primary visual cortex to learn how various cells responded to simple or complex visual stimuli. Lenses have been in use since antiquity. The first known lens was found in modern-day Iraq, and it dates back to approximately 750 BC. The ancient Greeks and Romans were known to use lenses for burning and for magnification. So, given the ingenuity of the ancient Greeks and Romans, it is somewhat surprising that eyewear did not appear in the historical record until 1286 AD. But lens technology continued to progress rather slowly, as another 3 centuries passed before the first compound telescopes and microscopes were invented in the Netherlands. The telescope, invented in 1608, was rapidly adopted by Galileo and Kepler, who refined its design. The compound microscope was invented around the same time and led to the discovery of cells by Robert Hooke. Even so, these early microscopes were of rather poor quality, as scientific principles of lens design and manufacturing were still not well developed. The compound microscopes of this era were so bad, in fact, that the simple spherical lenses of self-taught biologist Anton van Leeuwenhoek outperformed these early microscopes for many years. He kept his production technique secret so that he could essentially monopolize the field of microbiology for several decades. Nonetheless, the first use of a microscope in the operating theater was in 1686, when it was used to examine scar tissue and wounds. But it would be 2 more centuries before microscopes were used to assist with surgery. In the interim, significant advances were made in lens technology to correct chromatic aberration and other types of distortion. This entailed precisely combining materials with different refractive properties. In 1868, a physicist working for Carl Zeiss developed the groundbreaking apochromatic lens using new materials. He also put the design and manufacturing of lenses on a much firmer scientific foundation. Starting in the second half of the 19th century, magnification to assist with surgery began with the use of loupe spectacles. It was not until 1921 that the microscope finally made it into the operating room when Carl Nyland, an otolaryngologist in Stockholm, used it for otitis media. Otologists and ophthalmologists continued to innovate in their use of the operating microscope, and manufacturers made steady improvements. In 1952, Zeiss invented an operating microscope with a rotating Galilean device, adequate working distance, and coaxial lighting. This was incorporated into the OPMI-1. In 1956, foot and hand controls were developed for axial movements and image focus. The following year, in 1957, the operating microscope was first used in a neurosurgical operation by Ted Kersey at the University of Southern California for a facial nerve schwannoma in the temporal fossa, and the year after that, he clipped the first aneurysm using the operating microscope. He had been inspired by the famous otologist, William House. Shortly after Kersey, Pete Donahue began using the operating microscope and created a laboratory to study its use in Burlington, Vermont. His lab reported impressive feats of vascular surgery, but adoption was slow. One prominent critic stated that it's simply ridiculous to bring a microscope into the operating room. Later, in 1966, the young Dr. Yasser Gill came to his laboratory and developed new techniques for microneurosurgery, including the STA-MCA bypass, which he successfully performed after completing his fellowship. He also inspired many technical improvements to the operating microscope. For Yasser Gill, the stereoscopic perspective of the microscope was one of the major foundations of modern microneurosurgery, as it facilitates navigation through narrow corridors to reach deeper structures while minimizing unnecessary injury to the surrounding brain. In the years that followed, magnification and illumination improved, as did the mechanics, but the laws of physics placed limitations on the quality of imaging that can be accomplished through a pure optical pathway. For instance, as any photographer knows, increased aperture size allows for better illumination, but it also decreases the focal depth of field. With continued improvements in computing power and sensors, digital microscopes may provide enhanced visualization. Ideally, all parts of the surgical field should be perfectly illuminated, which can be achieved by increasing the intensity of light further, but that can actually damage delicate tissues, or by increasing the dynamic range of an electronic sensor. Alternatively, a series of images can be acquired with different exposures and then combined with a computer to achieve a perfectly exposed image, as seen above. Similarly, the focal plane becomes progressively more shallow at higher magnifications. The depth of the focal plane can be increased by combining images at different focal planes with software, circumventing the limitations of the optics. This section would not be complete without mentioning the endoscope, which has actually been in use in operating rooms since the late 19th century, longer than the operating microscope. But neurosurgical endoscopy did not begin to blossom until the 1990s. Currently, the field of skull-base surgery is being redeveloped with endoscopic techniques. The endoscope provides neurosurgeons new perspectives, but like the operating microscope, it requires the development of new instruments and techniques and appreciating new anatomical perspectives. As our ability to document the details of surgery and anatomic dissections improves, so does surgical education. The earliest written accounts of neuroanatomy are thought to have been produced in ancient Egypt and Memphis around 3000 BC, but these have been lost. Other scrolls, such as the Edwin Smith papyrus seen above, dating back to 1600 BC, describe trauma surgery and anatomic observations. And we have anatomical observations from Alexandria, of Herophilus and later Galen, which are preserved in Galen's texts. Yet, none of these ancient works include illustrations. This lack of images, as well as inaccuracies in Galen's observations related to his animal dissections — human dissection had been banned at that time — made the study of anatomy, an inherently visual subject, very difficult for over 1,000 years. The effective use of art in the science of anatomy finally occurred with Vesalius, who employed talented Renaissance artists to create images for his text, which were carved in wood block. The long-standing ban on human dissection had been lifted, and his text benefited from the invention of the printing press. For the first time, a high-quality illustrated anatomy textbook became accessible to a wide audience. He implored physicians to perform their own dissections, gently reminding them that the human cadaver should be the ultimate source of anatomical knowledge. Through direct observation of human cadavers, he was able to correct several of Galen's errors. Vesalius realized that anatomic knowledge is most effectively transmitted through a visual medium, so he went to great lengths to make sure that his scientific achievement was also a great work of art. His work therefore sat squarely at the crossroads of art and science, setting the standard for all anatomy texts to follow. Prior to this, Leonardo da Vinci had actually performed dissections and made enough illustrations to create the first high-quality anatomy text, but he never published it, and the illustrations were lost until the 19th century. Not to be outdone by da Vinci, Michelangelo was also very interested in depicting the human body accurately in his art, leading him to perform his own dissections. He considered illustrating a book of anatomy that would surpass Vesalius's work, but he never got around to it. Copper plates largely replaced wood cuttings later in the 16th century, allowing for finer tones and details. The lines of the drawing are engraved or acid etched directly into the metal. Shallow or more sparsely etched ink lines created the illusion of lighter shades, even though only one color of ink was used. Thus, anatomical images began to take on more realism. Over time, multiple plates were used to create color images. High-quality color images became more commonplace after the invention of lithography in 1798. A wax crayon drawing on limestone preferentially accepts hydrophobic ink versus the water-soaked stone. In the 19th century, Paris was at the forefront of lithography, and it had a thriving community of artists and anatomists, as well as hospitals that made cadavers readily available. So the conditions were right for art and anatomy to combine. In Leipzig, Christian Braun developed cross-sectional anatomy by slicing frozen cadavers and making vivid color lithographic illustrations of sagittal and axial cuts, which reimagined our modern MRI images. Ultimately, in the 20th century, metal plates with chemical coatings for photolithography replaced limestone once again, as they could be wrapped around the cylinders of the modern printing press. Interestingly, one of the best-known anatomy texts of the 19th century, Gray's Anatomy, did not aspire to the high level of artistic production that was made possible by the advancements of that era, but rather went back to woodblock carvings with a schematic quality, as the text was meant to be more affordable and widely distributed. Trained in Leipzig, Max Bradel immigrated to the United States and founded the first department of medical illustration at Johns Hopkins University while working with Harvey Cushing. One can see that the artistic techniques used for clinical illustrations continue to escalate in the 20th century, producing even more informative images. Harvey Cushing learned from Bradle, and he made a habit of documenting his cases with drawings in India ink and with photographs. As early as the turn of the 20th century, others became interested in using stereoscopic images for anatomy. In 1948, David Bassett, an anatomist working at Stanford University, began to collaborate with William Gruber, who had invented a popular stereoscope called the Viewmaster. Gruber photographed Bassett's dissections in color, and they finally completed a stereoscopic atlas in 1962 that contained more than 1,000 images formatted for the Viewmaster stereoscope. These atlases can still be found in medical libraries. Later, the neurosurgeon Al Roten Jr., who is considered to be the father of microneurosurgical anatomy, began using stereoscopic images to help residents understand three-dimensional anatomical relationships. As the operating microscope was being introduced in neurosurgery, he recognized the need to better understand the detailed anatomy seen through the scope, so he began performing dissections through the operating microscope that mimicked the view from a neurosurgical approach, while revealing the relevant anatomy in much better detail than an intraoperative view or a typical cadaveric prosection. Dr. Roten also pioneered the use of dual projector systems with polarizing filters to give stereoscopic anatomy lectures at courses and conferences. These lectures were ultimately reproduced in several 3D formats for the Roten collection. We are now moving beyond stereoscopy into an era of photogrammetry, which is a process where photographs taken from a variety of perspectives are combined to create a 3D model that can be manipulated on a computer or in a virtual reality setting. Moreover, similar technology allows artistic illustrations to move from flat images to three-dimensional models that can be rotated and peeled away in layers. This allows one to learn anatomy in a more interactive fashion and to rehearse surgery. In just over a century since the first moving pictures, and just a few decades after a time when video editing was prohibitively expensive, we can now produce videos of great sophistication on an inexpensive laptop computer. With a wide availability of video streaming, over the past decade, we've created vast libraries of surgical footage that are at the fingertips of neurosurgical residents, allowing them to learn from surgeons all over the world at an accelerated pace. I am sure that early neurosurgeons would be amazed by all of these advancements. And the artists and physicians of the Renaissance, if they were here, would agree that we too are in the midst of another Renaissance that will accelerate our craft faster than ever before through improved visualization.